The Sargent family, dear friends and colleagues, a very warm welcome to the 31st Sargent Lecture. It gives me great pleasure to welcome today's lecturer, Professor Nilay Shah. I'm sure Nilay is known to many of you, but let me say a few things about his career. So Nilay got his first degree from Imperial and also his PhD from Imperial under the supervision of Roger Sargent and Kostas Pantelidis. He started in scheduling, supply chain, process synthesis, so with some very fundamental PSC challenges. But his range of expertise and his contributions is really staggering. He's contributing to energy systems from the small scale of process units to the scale of infrastructure of energy systems, but also making significant contributions to vaccine manufacturing, as we'll hear about today. He's really sought out for his expertise in all these diverse fields. He's a member of many an advisory group, including the DESNES Science Expert Group, the Hydrogen Delivery Council, the Low Carbon Hydrogen Standards Working Group, the Hydrogen Offtake Working Group, and the Government Vaccine Development Working Group. So you can see that Nilay is making an impact much beyond academia in shaping the future of the world and of the UK. He's also sought out for his broader leadership and his advice. I think systems thinking is about bringing clarity to very complex issues. And Nilay has been head of department of the Department of Chemical Engineering here at Imperial. He's been the director of the center, but he's also been on the ethics committee of the Royal Academy of Engineering, on the diversity committee, and he's chaired the history group at Imperial. So really making an impact also on broader, more complex issues. He's been recognized for his work in many ways, with an OBE in the Queen's Honor List. He's a fellow of the Royal Academy of Engineering. He's been elected to the American Academy of Arts and Sciences. He's received the Sargent Medal, the Sharma Medal, and many, many more awards. So we're really honored, Nilay, that you're delivering tonight's lecture, and really look forward to your talk. Thank you.
Great, well, thanks very much. It's very strange to be standing here now because I've been in the audience for these lectures for about 30 years. So here we go. When I started, this had 150 slides, my first draft. So you can see it's only got 56 now, so you should be happy. So first I want to say a few words about Roger Sargent, because I think many of you know who he is, but some of you, he might be a less familiar person. So, this is a saying from one of our many colleagues, that he was a true gentleman, above all a very kind human being. But of course, we know him very much also for his contributions to chemical engineering and process systems engineering. So, potted biography. He grew up in Bedford, went to school in Bedford, studied, like me, at Imperial College, undergrad and chem-eng, went to work in France for a few years with a young family, came back to Imperial and started as a professor. For a very, very long time, he was the youngest professor in chemical engineering, person made professor in chemical engineering here. And he participated in many other things as well, like what was then the City and Guilds Institute, which is now approximately the Faculty of Engineering. He was a department head, surprisingly, for 13 years. Omar, you've got a way to go yet to catch up with him. And he was a founding fellow of the Royal Academy of Engineering, or Fellowship of Engineering, as it was first called. And perhaps most importantly, in the context of today, he had the vision, along with some collaborators, to establish the Center for Process Systems Engineering. The center was funded by a special program that the UK government launched, which was to set up large interdisciplinary research centers in topical areas. And there are only two such things still existing from that original cohort from 30 years ago. Ours, which I hope you agree with me is thriving, and a very, very influential one in optoelectronics in Southampton University. So Roger, he was really great fun to work with. He was a very, very clear thinker. I think that's why he sort of established this whole concept of process systems engineering, is because he had a very interesting mind, in the sense he could conceptualize things at a very abstract level, but in a very influential level, and then just write them down in one go. This is one example of some notes he shared with us. It was relating to a student who was working in an area similar to me. So he had this idea, and he just sort of wrote it all down, all in one go. And this is how he used to work, and the students would go away and try out these concepts. Not only was he a very clear thinker, but I think one thing I really found interesting about him was he was always optimistic about how far you could push the boundaries of technology, of ideas, of algorithms, and so on. And we always used to say, if you wanted to summarize him, it'll be all right on the night. And this is one funny example, which was that this was in the very early 90s. So there was this board you could put into a computer which contained these processors called transputers. So in theory, they could solve things in parallel. So it was like the maybe great-grandfather of HPC today. And of course, he had this idea of how you could take some complex algorithms and just parallelize them and solve them. And of course, he had a view, first of all, that someone like me, and I was probably 23, I guess then, something like that, when he asked me, go and look at this. This is the idea. This is the tool. Just put it in the computer. This is the technical note, and I'm sure it works. So first of all, I think he's very optimistic about the technology. He's very optimistic about what I could realistically do. We got bits and pieces to work, but it didn't really work. The other thing was, he had a very wry sense of humor. And one of the things he found very funny was for my predecessor, Amelia, one of his PhD students. She was working on scheduling of batch processes. And she needed some examples, and he had a lot of experience of industry. So he came up with this so-called fine chemicals process to make a relatively small volume product, maybe a pharmaceutical intermediate. And it's quite complicated. So the rectangles are processes or tasks, and the circles are materials or states. Anyway, it was very interesting, and she used it, she published it, and it became referenced a lot. And what you'll find is if you read the scheduling literature, so many people use this example, and they refer to it as the fine chemicals process. And I remember he used to find it really funny, because it was something he just completely made up. It's no real process, but every subsequent paper seemed to think that it was a real thing. And extremely timely. I don't know how many of you have seen the Chemical Engineer for this week, but they're running a series of articles called My Hero. And David Simmons, who may even be in the audience here today, hopefully I know he was planning to come, he wrote an article for them, which was, my chemical engineer here is Roger Sargent. And so I definitely recommend you to read this article, because it says a lot more about him and his achievements. But also it's very much a personal reflection from David. So yes, fortunate that it's literally just come out. Okay, so vaccines. Why did I choose this topic? I thought it would be quite fun to talk about vaccines because, most of us, we don't sort of use hydrogen, or we don't, get our hands on ethylene or anything like that. So although those are really interesting things to talk about, how you make them and how you use them and so on, well, we all have experience with vaccines. We all use vaccines regularly. We probably don't think about them very much. And here I'm quite interested in the fact that actually they're quite complex materials, and there are a lot of interesting systems types of problems that apply to vaccine manufacturing. So I thought I probably should start with some introductory material. It might be all obvious to you, but vaccine is a biological material. It's something that's quite strange from the point of view that it ought not to be particularly controversial, but it is controversial, and it's controversial because it's something that you take when you're well, and it's a preparatory thing, so it's prophylactic. And I think human beings are not good at being proactive about things. We're good at reacting. So if someone gives you a diagnosis, you're really ill, but if you take this antibody, you have a really good chance of recovery. You're not going to worry about all sorts of things like, is it safe, and are there side effects, or whatever. You're ill, you're going to react to the circumstance, you'll accept to take the antibody, and you'll hope for a good outcome. Vaccines have this controversy, but they're not particularly things that should be having this controversy. So they're biological material. You can take them in different formats, and they're meant to stimulate an immune response in terms of your cells, certain cells and antibodies. Lots of different forms. So sometimes it's the actual pathogen that makes you unwell that is modified in some way, so it won't actually make you unwell, but you'll get the correct immune response. Sometimes it's an element of the pathogen, like a surface protein, for example. And interestingly enough, two new modalities, and one I'll spend quite a bit of time talking about, is messenger RNA or self-amplifying RNA, which essentially is a code. It's like a digital code. It tells the cells in your body to make certain proteins, and those proteins are displayed in a way that looks a bit like a virus, and it gives you the same immune response. And viral vectors have a similar kind of concept, but they're packaging the genetic code in a different way. They're using a virus that's not going to make you unwell, but it's going to get the genetic material into the cells. So that's different kinds of vaccines. How about how they're made? So depending on the type of vaccine, the way you make it is also different. So particularly the ones that are the actual pathogen, you have to culture it in an environment, which could be cells or eggs. Otherwise, if it's just a protein, then you can produce it using an organism like a bacteria or yeast, which you genetically modify, and it produces that protein, and then you need to separate that protein from everything else, formulate it. You can actually then also not just have that protein as a protein, but you could have a number of components that are like those proteins, and they self-assemble to make a structure that looks a little bit like a virus and gives the immune response of a virus, but it isn't a virus. It's called virus-like particle. mRNA. So now, this is, in a sense, an advanced technology. You're instructing your own body to make the actual biological element that in turn provides the immune response. So you give instructions to the cell in the form of messenger RNA to make a specific part of the virus. So for SARS-CoV-2, that's the spike protein. To make sure that it actually does the job, you need to package the RNA in a lipid nanoparticle. So that then has two effects. First of all, it allows it to be transported and stored. But secondly, when you have an injection, it will pass through the cell wall and create start the translation process. Viral vectors are quite similar sort of effect, again, to get that genetic material into the cells. And then another type of vaccine manufacturing is down to membrane vesicles. So what they are is particularly from bacteria. The cell wall forms these things called blebs, and they bud off the cell, and they look like mini versions of the cell. So they display very similar properties. They're smaller versions of the cell. Of course, they're not real living things. They won't make you unwell. But again, they can give you the right immune response. So let's look at it from the point of view of the kind of research we do in the Sargent Center. So if we look at biotechnology generally, and then just think of vaccines as a special case, it fits the paradigm that we often like to show of multi-scale modeling, where we've got some things which are happening, at the very fine scale, where we might be trying to understand, for example, the genetic code for a specific protein and optimize that to get the right protein structure, and then to think about how we might manufacture that, for example, in a process, which could be a fermentation or something that we call in vitro transcription, which I'll talk a bit more about. And then how do we integrate that into an overall system, upstream, which is manufacturing your basic material, downstream, purifying it, and then secondary, formulation and distribution. Then, of course, we're often very interested in things like supply chains and distribution. How do we actually get this to where it's needed? And increasingly, now people are talking about sustainability. So life cycle analysis. Historically, we've always thought about that for big bulk materials, for energy, water, food. But increasingly, pharmaceutical companies are having a strong focus on sustainability as well. So I've been working things related to vaccines for about 20-ish years, 22 years, something like that. And, just to bring it back to the sort of PSE world, what kind of things have been involved, and we'll see in the rest of the talk, things like scheduling and manufacturing optimization, dynamic systems analysis, which I won't talk about today, where we're trying to model disease progression and vaccination together. And then a lot of process design using what are the sort of classical techniques that are now becoming quite embedded in a discipline called systems-based pharma. So I'll illustrate these methods by some examples. So I'll start with the first problem, something we were working on quite a long time ago, which was influenza vaccination. So I hope many of you have taken influenza vaccines. They have two benefits. First of all, you're much less likely to get a bad form of influenza, but you'll be surprised to hear that people who have historically taken influenza vaccines regularly will be much less likely to have strokes, heart attacks, various other diseases that don't sound like influenza, but they result from, let's say, some effect of repeated illnesses. So how do you make influenza vaccines? There are two ways, but I'll focus on one. Well, there are some new ways coming through as well. So the egg-based production process probably sounds quite strange to you. And here are some actual pictures of the actual eggs that are used in these processes. So these eggs are actually fertilized eggs, so they act as mini bioreactors. They're like mini cell-based manufacturing environments. And what we do is we inoculate the eggs. We then let them grow the virus for a certain amount of time. Then we need to harvest the virus from the eggs, purify it, inactivate it, formulate it, and ship it. Quite a complex process, and we'll see a bit more how complex it really is. The other thing that's complicated about this is that what happens is you have viruses circulating around the world. So we all, are familiar with having our influenza vaccination around September or October through to maybe about January. So what happens is there's a sort of surveillance process, and it particularly starts to look at what's going on in, for example, in the southern hemisphere, which viruses are in circulation, and usually there's one strain. Robin will remind me what it is, but I think it's H1N1 that nearly always is included in the vaccination. But when you go for vaccination, you're actually getting either 3 or 4 flu vaccines in one. And what this surveillance is trying to do is to estimate, based on all the information possible, what are the 4 most likely strains of flu that will be circulating in the winter. And so as soon as that's known, it's almost like a starting gun is fired. So once you've got that data, you start producing as quickly as possible. Because you often see things that the vaccine has run out, and that's because it actually takes a long time to make these vaccines, but you can't start earlier than around March because you don't know what you should be making. So we were working with a company that operates this kind of process, trying to model the process and looking at the fact that demand is growing. Yeah, we've got aging populations in the northern hemisphere, and there's more encouragement of uptake of flu vaccines. So demand kept growing, and what was a comfortable manufacturing process was getting very uncomfortable. So this is what it looks like. Again, as I say, you've got your, you prepare your vaccines, you inoculate the eggs, you incubate for a certain period, you inspect, harvest, and then there's a box here called downstream process. This is a trivalent vaccine, so we get 3 bulk, what are called monovalent bulks, A, B, and C. We pool them, and then we fill them into vials, and then there's a few other quality control things, and then we've got the final product. So this downstream process, so for those of you that work in biotechnology, you'll see this looks really familiar, lots of familiar unit operations, filtration, adsorption, and dilution, and so on. So a lot of the familiar kind of processes, some chromatography as well. And eventually through these many, many steps with many, many, many reagents, we end up with these monovalent bulks. So the question we were asked is, please optimize this facility so we can produce a lot more stuff. And of course, we looked at it, and you can see the complexity. This problem is too difficult, well, it was then at least, to solve within one optimization model. So we had to decompose the process. We saw there were certain places where they were keeping buffers of material, so you could decompose the process, and we developed a kind of fancy heuristic for one part of the process, but we were able to use optimization with another part of the process. We were able to build schedules that look like this. So, down this side here are the different pieces of equipment, and here's your time scale. Again, February, this one is able to start February through to September, October, and you can then see that you can see how much you can make by the time you want to be getting those vaccinations out. There's a color coding. So I told you there are 3 different vaccines. So you've got your light orange, your dark orange, and your yellow, and that's just showing what's in production at the time. That's the upstream part, and then we've got the downstream and filling part. And so, this is showing that you're starting to get significant amounts of actual useful products coming through by sort of June. You can start building up stocks, and it's already being shipped out. So, what we found was that there's incubation, which is the eggs just sitting there with the virus replicating in it is the rate determining step. And one of the other challenges is that to make a fully formed vaccine, you need the dark yellow, the light yellow, the dark orange, the light orange, and the yellow. So you need them all to be ready, and then you can put them together and make the vaccine. But if you mix them all up too much, it gets very complicated for how you run the process, because they're not necessarily compatible with each other. So you don't want too much mixing up. So you can see here, they're happy to run the light and red orange kind of together in the facility, but the yellow, they want to keep it separate. So you don't want to, you want to make sure you've got clear segregation and you know what you're making. And so there's a tension between getting early batches of all 3, because obviously if you make A, B, C, A, B, C, A, B, C, you'll have it earlier, but it's less efficient because it ends up being much more complicated. So, what they found was they were predicting the future demand for this facility, and it was very difficult to meet that. So it was interesting because this facility was in a former communist country, and they had a very specific work schedule, because this is probably maybe, it's a very high quality operation that they had there. I mean, it was really, really impressive, and it was bought by a big multinational. So that is this interesting thing that they operated 2 shifts of making, and then 1 shift of just completely cleaning the whole facility. So it was like a 16+8 kind of system, and that was slightly staggered between the different unit operations. So we sort of worked with them to look at the actual activities involved in cleaning and whether you could actually do a 3-hour cleaning and then have some production as well rather than that 8 hours. So we were looking at some things like add some more incubators because that's the bottleneck, or look at a different operating schedule. And so we ran these through the optimization. This is just indicating with the pictures that the incubators are really heavily utilized. Inactivation can also be quite busy too. And so what we found was, the base case was 67 batches over a certain period. B was shortening the cleaning time, which gets us up to 93 batches, and then adding some additional units can get you up to 98 batches. So you can start to see that, you could have about a 30% increase in production by changing the way you do cleaning, and you could increase production even more by adding 1 more incubator. And once you do that, you're starting to make the inactivation the bottleneck. So they went away, and apparently they did rethink the way they did cleaning. So they didn't have any tools like this, so using these process systems tools really made a big impact in the ability for them to increase the production of influenza vaccines without necessarily investing in new capacity, just by rethinking the operations of the plant. So moving up from production to looking at an enterprise as a whole, or multiple enterprises and supply chains for vaccines, the next problem we started to look at was what we call capacity planning. So now we're thinking about how do we meet the growing demand for vaccines. Okay, we can squeeze a bit more out of our existing facilities, but eventually what we need to do is invest in new technologies that are emerging and new production facilities. And so, there's lots and lots of demand. This was even from a while ago. So there's, billions of doses of vaccines a year. And these are just your primarily regular pediatric and geriatric and other vaccines. So let alone some of the things that are coming through nowadays. And this is just showing, what people were planning at certain times. So, people were making a lot of vaccines already, So the rows are different, some of the different manufacturers, and the columns are some of the vaccines that were being produced or in development. But this was just a snapshot in time, so it just shows you that people are constantly innovating and developing new vaccines. So some of the big new vaccines that people are developing now are for shingles, for example. So that is something that is not even on this list. So there's a constant level of innovation. And what that means, of course, is that you need to think about where you're going to make those vaccines. Are they going to be in your existing facilities? Are you going to build new facilities? Now, the problem with vaccines and many pharmaceutical and biological products is you don't really know how much you're going to need, especially if you've got products in clinical trials. So if you go up here, some of these things, the rotavirus, for example, the multivalent meningitis was then, RSV, these were things that are in clinical trials. And you might be monitoring the data that's coming through. But from that data, you've got to start estimating how much I'm actually going to need to make. And often in clinical trials, you're trying different dose regimes as well, so you don't even know what the dosage is going to be per person. So we end up with a lot of uncertainty. So, this got us to think a lot about how we optimize manufacturing under uncertainty. So think about uncertainty with different levels, qualitatively. So you've got an existing product, an existing market. So influenza in the UK, you know the population of over 60s, you can predict. You can look at last year, and this year is going to be much the same. Existing product in a new market, so you want to take an influenza vaccine maybe to a developing country context. So you can look at maybe who the purchasers are. Is it going to be a national health service type thing, private? So probably medium. So then launching a new product in an existing market, so something like RSV or shingles, again, you can do some demographics and various other things, look at producer costs and purchaser likely to pay and so on. You're starting to medium to high. And then you've got clinical trials, new products for new markets, high uncertainty. So what made us think about this project was a thing that happened in Bayer, which was they were developing a medicine for toxic shock syndrome. And they were really confident about how it was going to go. And so they invested in a very specific facility, which was designed particularly for manufacturing this product, even though it was in phase 3 clinical trials. But the results were coming through very good. So they spent about $200 million as it was then. It'd be a lot more now. But actually, as data were coming through, it suddenly came through. There were a lot of side effects. So they ended up with a very nice facility, but it was inappropriate for the product because the product was not going to be there. So if you build capacity ahead of demand, risk of wasted capital. However, we also saw an alternative thing. This is way back when we were doing these studies. For many indications, we saw shortages. But the one that comes to mind is actually Viagra. So you wait for the demand, and then you're always playing catch up. And of course, we saw this with COVID. So you have a loss of market, or even worse would be adverse health outcomes, as we saw with COVID, with some parts of the world waiting a very, very long time to get access to vaccines. So how do you balance these tensions? Well, obviously there's commercial arrangements that can help balance the tensions by underwriting, and that can say, look, we'll guarantee to buy X numbers of doses, so please go and build the facility. But what we were looking at is scientifically, how do you balance this? So here, we're trying to do capacity planning, where we know what a firm is making today and how those will grow, what's already ready to go into manufacturing, what things are in various stages of R&D and clinical development. And so we've got that data, we've got forecasted demands, we've got the existing technology, some of the new technologies, particularly when we were doing this work, it was mainly the mammalian cell-based type of processes and related ones. And so we were starting to build a model. The model would also look at where are the R&D products, for example, in their different clinical trial stages, and the company we're working with had this specific way of modeling what happens in clinical trials, where you've got failure or low success, which means you're very likely to launch this product. Low success means it sort of works, but it's not got the efficacy really that you want. You've got target efficacy and then high success. And that's in different stages of development. And so, we can also consider that this product one is nearly known, but we've got other products in the horizon down in the future. So we're looking at facilities that are making multiple products. So we solve this with what we call a stochastic optimization framework. It's a framework where we capture these uncertainties in this scenario tree. And what we are trying to determine is essentially where you can expand capacity in existing facilities, where you would build new facilities. There's some secondary things that come into play, like there are different geographies where you can put these facilities, and the tax regimes and incentives are different. But the primary thing is you don't know exactly what you're going to sell, but you can optimize balancing across the uncertainties. So one of the things you can look at is if you use net present value as a way of thinking whether investment makes sense or not, you don't get a single net present value, you get a distribution because you're uncertain what can be sold. So, this is an example where the average or expected net present value is about 3 billion units. There's a probability of losing money of 2.7%. In other words, net present value is below zero. If things go really, really badly, you would lose 288 million. And if things go really well, 6.7 billion. So you can show this to someone, and they just look at that and think, that's not particularly useful. It doesn't really help me with my strategy. So, you've done this really nice analysis of all the products in our portfolio, all the things in trials, all the things in R&D, and you've given me a nice chart to look at, but it really doesn't mean anything. So we had to do a much deeper dive. And so, we were looking at what actually happens in these cases. So what we can see is you have lots of possible outcomes. So for example, the so-called production time, it's an approximation of how much capacity you need. So if you're very unlucky, you won't need much capacity in the future. You'll be down here. If things go really well, you'll be up here. You need a lot of capacity, and there's a lot of other things in between. And these are just some examples, like a bad case, a medium case, and a really good case, just showing you outcomes. And if you're really clever and you know which one you're on, you can see how much capacity you'll utilize. But this started to give us some ideas of how we actually design these vaccine manufacturing facilities. And so we spent a lot more time then with the engineers that actually designed these things. And we came up with two strategies for doing this in a bit more sophisticated way, rather than the classical, how do we optimize things under uncertainty that we do in PSC. So what we're trying to do, of course, is we're trying to balance this issue of don't wait too long, because even if you see something successful, it's going to take you 2 years to build a facility. But don't build things if you don't know what you're doing either. And so we came up with two strategies. So the first one is, okay, you acknowledge all the uncertainty, and then you try to get the best possible decision today. Like, we're just going to make all our decisions today, and we're going to decide where to build things, how big they'll be, and then we'll see what the future brings, which we call strategy B. We make all our decisions here and now. The alternative is what we do is we are going to go for strategy A, which is we're going to design a facility, but we're going to keep the facility separate from the number of production suites or production lines in that facility. But we'll design the facility in such a way, in terms of air handling, water for injection, utilities, and other things, so that you could quickly add manufacturing capacity within that facility. Now, the one thing, of course, you need to do in strategy A is you need to think up front, what's the maximum number of production suites I will have in that facility? But it's obviously a much lower investment up front, and then you can see how well things are going. So this is just showing how these strategies work. And so what happens here is, if you commit to relatively few facilities up front, like 2, which is this line here, you're guaranteed to make £1 billion net present value, because you build this relatively small facility, fill it with 2 suites, and you decide that probably, you probably don't expand capacity much. So you'll fill it, and you'll be happy making money. Of course, what you're also doing is you're probably missing out on, other revenue. But what happens here is you can see that once you reach around, looking at these colors, probably around 16, so you size something that's for 16 suites, or production lines you can think of, then you will take up those 16. But if you sized it for 20, you would never actually take up all 20. That's why these lines are starting to converge. This is the case where you fix everything up front. So when you fix 2 or 3 and you just build the whole thing with 2 or 3 things in it up front, you get this line here, a bit like what we saw before. So this kind of pink color and this brown color are the same. However, let's say we go crazy and we build something with 26, huge factory, 26 production lines. We build it all up front. We get this very big distribution. So this is the net present value, and this is the cumulative distribution. So we might end up with, losing $6 billion. We might make $6 billion and everything in between. So if you follow this pink line, it's your extremely aggressive, optimistic kind of strategy. Build it and they'll come kind of strategy. So build everything up front or build the infrastructure and then fill it over time. This is how the distribution goes. And so, this started making us really look at some of the financial literature and think much more about how do you do optimization? How do you measure which one is better? And we realized, that there are a lot of interesting measures that people use for looking at these distributions. So here are two possible distributions. Again, blue one is like a conservative one, and red one is a more aggressive one. And we realized that there are many measures people use. So there's the expected net present value, there's the expected downside risk, the worst case, the best case, various things. And you can start to use these to make people think a bit more about how risky they want to be and start to have a proper strategy for how you invest in future manufacturing facility capacity. So let's start looking at how this can help you. So here, strategy A is in red and strategy B is in blue. So this is the original model now, again, where expected net present value is just your metric. And this sort of tells you that if you go for strategy B, then you should build something, if you're only worried about expected net present value, capacity about 10 to 12, and stop there. Whereas if you go for strategy A, you can go a bit bigger, but you may not fill it. But if you build something that's too big, with strategy B, you start doing worse because you've spent a lot of money and you never recover that capital because the demand isn't there. And that's the expected. That's taking the average over all possible outcomes. If your decision criterion is the so-called maximax criterion, so this is, as I say, there are all these different criteria. So maximax is the way the optimist would think about net present value. That all our drugs are going to be really successful and they're going to be really efficacious, so there'll be huge demand. Therefore, I'm going to pick the scenario on my net present value distribution that's the best. Then you'll find that you really want to go large. So even in strategy B, where you do all up front, you go for a 22 size facility. Of course, it's a very, very risky one, as we'll see in a minute. If you're a pessimist, which is the other way of looking at it, the so-called maximin view, which is please make a decision that I will, even in the worst case, have a decent net present value, then essentially you probably wouldn't do anything. You'd say, oh, well, we might even build a very small facility, but we might be really unlucky, and a competitor will take the market share of the existing ones, and all the ones in trials are going to be unsuccessful. And so, yes, you might go for a strategy A, but you probably wouldn't do much for manufacturing. There's a funny decision-making criterion on uncertainty. Well, I find it funny because it's mainly psychological, and I think actually it relates a lot to how we make decisions when we're dealing with uncertainty, which is this quite strange concept of regret, which is if I make a decision today and I commit to it, how much better could it have been? So could I have made another decision that would have been much better? And what this regret thing, which is often used in economics and in politics, it's really looking at, I don't want to do anything too stupid, is how you could translate it. Because I don't want to feel in a year's time bad about the decision I've made. So here, what we're trying to do is we're trying to minimize the maximum regret. In other words, I want to minimize my feeling of disappointment that I didn't take the right choice. And so what happens here is it shows you that in this criterion that if you're going down B, you want to go to about 10, and that allows you to feel that I will, in both cases of things went worse than I expected, or, oh goodness me, we've got all this unfulfilled demand. In both cases, you know, the value of that disappointment is about 4 billion. And the nice thing about this strategy A is as you make the facility bigger, because you can fill it over time, you have much less regret because you haven't committed all your choices up front. Because you can think, okay, I have some regret if I'm over here, in the sense, you know, I went for 12, I filled, you know, maybe I'm in year 6 or something, I filled all those 12, I should have made it bigger, damn, you know, and then I would have made 2 billion more. But it's still a good decision. So that's, you know, just a nice way to think about uncertainty. What's funny about all of these things, of course, is we still don't have an answer for the client. We've just got more graphs to show them and more things for them to think about. And here's when you start looking at it in 2 dimensions. So we have another metric called expected downside risk, which is essentially, you know, the combination of the likelihood and the magnitude of the NPV falling below a target level. And this shows you now, you can see strategy A performs very well. So on the x-axis is the expected net present value, so how much money you on average will make, and the y-axis is a measure of risk. So as you go up the y-axis, it's becoming more risky. So this is 2, 3, 2, 4, 6, all the way to 26. So essentially, you know, this is a really stupid decision, 26 size facility with strategy B. It's high risk and low reward, so you really don't want that. So it's interesting, because normally with a risk-reward system, you always get more reward by putting more risk, but here it starts to invert. But on strategy A, you can actually have a good reward with a low level of risk. So now you can start making decisions. And then again, there's another metric called value at risk. Value at risk is often quantified at a 99% or a 95%. So it shows, you know, with a 95% certainty or below a 1% certainty, how much might you lose? And so here again, you can see that as you accept more risk, you can get more expected return with a nice frontier, whereas with strategy B, you get this funny shape again. So you wouldn't, again, you know, the one conclusion this tells you, if you're going down strategy B, don't go for more than about size 10. If you're going for strategy A, you can be quite a lot more aggressive. So funnily enough, we were discussing this work, which happened quite a long time ago, in a new project we're doing with one of our consortium members, actually looking at energy storage systems, where you want to size your energy storage system, and you're going to buy cheap electricity and sell expensive electricity. And obviously, if you make it huge, what happens is when you're trying to sell the electricity, you perturb the electricity market, because people don't want that much, so you can only sell it at a cheap price. So you start getting this inversion effect again. So it's kind of an unusual effect, because normally you expect to see a risk-return profile that is just monotonic. So let me come to third problem. How are we doing for time? Not too bad. So we're now going into zooming down from production scheduling and capacity planning, zooming right into process design. And this really was something that we started off with Robin, who's in the audience as well, where we started thinking about the fact that, you know, we really need advanced and quick and responsive and flexible manufacturing facilities for vaccines. Because although I showed you that picture showing what the companies want to work on, rather sadly, in 2018, we went to a funding panel with a hypothesis that it's only going to be a matter of time when a so-called disease X will occur, and we'll need a quick pandemic response, and we need a platform that is disease agnostic. So we started, this future vaccine manufacturing hub in 2018, where we're looking at this concept of platform technology. So the idea here is that you, first of all, bring a lot of systems thinking into the design of these platforms, and you make sure that they're disease agnostic and that they're rapidly able to produce products because you understand the process from A to Z, and that's where the systems-based pharma approach comes in. Well, we're working on 4 platforms: RNA platform, a VLP platform, a humanized yeast platform originally, and what's called a custom outer membrane vesicle. Now we've added an insect cell platform to that as well. But I'm going to focus on RNA because probably almost everybody here has had an RNA vaccination. When we were pitching this to the panel, they were quite interested in this very novel concept of an RNA vaccine. And we said, well, I remember someone asking us, when do you think someone will actually have one? And I think we said probably within about 8 years they'll be available as regular vaccines. So the way it works, this is our vaccine here, right? It's encapsulated in this nanoparticle, which allows it to be released into the cell. Within the cell, then, you have this process of translation that actually produces a protein sequence, which then conforms into the correct structure. For example, the COVID spike protein, and then that's displayed, and that's what starts the immune response. So this vaccine and this process, it may last a few hours, few days, maybe a few weeks, depending on the type of vaccine, and then that RNA is gone, but the immune response remains. So RNA is kind of complicated, but the important bit for you to worry about here is the fact that we have this blue bit in the middle, and that is just a genetic code. So it's a 4-letter sequence. Could be maybe tens to hundreds of bases for something that's not for a vaccine but for another purpose, and typically several thousand for a vaccine. For certain vaccines, we have an interesting feature which is called a self-amplifying feature. So that is an additional bit of code, and it instructs the body not only to make the protein, but to make copies of the RNA. So you can have a much smaller vaccination dose because your cells will make the same RNA as well. So these are produced not by a direct biological process like fermentation, but actually by essentially a chemical process. So in the chemical process, we have a in vitro transcription, which essentially requires us to provide a DNA template, and from that DNA template and the precursor materials, you have an enzymatic process that creates the RNA. So it just makes lots and lots of complementary copies of the DNA, and hopefully makes it, first of all, what we call capped. So you can see here we have these caps here. This ensures stability and effective operation in the body. We also have a tail, and this tail also helps us a lot with the separation process. So we make the RNA, we have to digest that DNA template. We then have various separation steps, and then we might also have the various filling and so on, and then it's ready to go. So one of the concepts that we've developed, and my colleague Cleo has been very much involved in this, as well as my colleague Maria, is a process for applying the so-called systems-based pharma to products that are at a very early stage. So systems-based pharma began in the small molecule world, where it's chemical synthesis, it's unit operations like filtration, reaction, crystallization, and a lot of historical operating experience. So now what we're trying to do is we're trying to take the same approach and rapidly apply it to something that still has a lot of development and uncertainty. So, looking at similar products, patent databases, scientific literature, to allow us to start to understand what are the important features of this system. And what's nice about the systems-based pharma approach is it has a very structured methodology. It makes you think about three main things. Primarily, and most importantly, the so-called critical quality attributes of the drug. In other words, what are the properties of this drug that you can measure and you can put constraints on, so you can say, is it going to work? Is it going to be acceptable? Are we going to allow it to be used? So the first thing you have to do then is decide what those are qualitatively, and then quantitatively set the limits on it. Second thing is the critical process parameters. Essentially, what are your adjustable parameters in your manufacturing process? pH, concentration, temperature, time, all those classical things. And then, of course, you have to work out how those process parameters affect the quality attributes of your product. There's a third thing called the critical material attributes, because you're also using ingredients to make your product, and how pure those are will influence how good the quality of a product is. And of course, you don't want to spend more money on those ingredients than you should, so you need a good understanding of how those critical material attributes also affect the critical quality attributes, and of course, there's often a feedback with those process parameters. So that's the systems-based approach, and that is exactly what is used in this systems-based pharma technique, and it's embedded in a process called quality by design, often how we call it quality by digital design, where we, work out the patient needs. So that's usually the dose regime and something about the quality. This is a purity profile. This allows us to identify those CQA, CPPs. We start to understand how they influence each other. So you can see this is where modeling comes in. And that gives us a so-called design space, which is how we will operate the process to make sure it will always produce quality products. And so, we can start to identify what those are and which ones are important, and then we can start qualitatively and then quantitatively work out how these parameters affect the qualities. That then allows us to go into experimentation, because having done that semi-quantitative analysis, we can say, these are the things we really need to measure in experiments, so we can target our experimentation. And often, what we'll do is we'll quite quickly try to develop mechanistic models. We have to take Box's famous saying that all models are wrong, but some are useful. This is definitely an example, but it allows us to do quick exercise in terms of thinking what matters, what doesn't matter, and how we're going to design experiments. Making RNA requires very expensive reagents. It takes quite a lot of time of people in the lab. So it's not a thing for doing super high throughput screening. It's a thing to apply some thinking to. Let's just put that. And so you can start doing preliminary experiments to simple models and build an idea of what we call the design space. So for example, here we're showing your NTPs, which are your primary reagents to the individual components of the RNA, the 4 types of nucleotide phosphates, the magnesium, which is very important, and the enzyme called T7 RNA polymerase. So this shows you might want to operate in this space, but actually that's an expensive space to operate. So that approach has been encapsulated in a piece of work by our PhD student Simon in an overall workflow, which actually allows us to go from the principle of a new disease has come in, we've sequenced it, we're getting some idea of what we want to make, and it allows us then to have a platform that's ready, because we've actually developed a design concept for the platform that's independent of the disease. So we only need to do a bit of disease-based analysis, and we can go into manufacturing. So there's something called the 100-day challenge, and it's essentially saying, how can we establish some principles and manufacturing technologies and standard operating procedures so that if there's a disease outbreak, we will have a vaccine in 100 days. And this is really the way to do it. So coming to this RNA in vitro transcription process, what we find that a lot of people are doing a lot of experiments all around the world, and there's a lot of variability in some of the main KPIs. So economically important is the titer. So you tend to have small reactors, few liters up to maybe tens of liters are the biggest reactors. And you can make of order a few grams per liter, but you can see a big variation in there. There's the integrity. So how much of this RNA is full length, properly capped, good quality RNA? It could be anything from, in some cases, 50, 60% up to the 90%. One problem we have with an RNA is when you make RNA, you often end up also with double-stranded RNA, and the mechanisms for this are still being explored. So that's something where, instead of one linear, long, 1,000+ unit molecule, you've got these double-stranded, and because of the complementarity that you get between the bases, this double-stranded RNA is quite stable, and it's not a thing you want in your actual drug product. The other thing we find is when you look at how people do these experiments, and these are some of the critical process parameters, these are your process variables, they operate in a very, very wide range, like very dilute, very concentrated, and everything in between. And a lot of these things, as I say, are very expensive, so you don't want to use more than you should. So there's a kind of big rush to do research in RNA manufacturing, but it's quite empirical. And so what we've been trying to do is bring a lot of systems thinking. And what we're trying to do is understand what are some of the fundamental mechanisms for how RNA grows and is produced, and what are some of the things that can also go wrong. So this is how it works. So this is our template, and this is the enzyme. This is the RNA growing on the enzyme. Sometimes we get this abortive initiation. So after a few bases, it comes off, and so we see these in solution. But here, it's growing nicely, and it's going well. So this is the elongation phase, and then we've got full-length RNA. So we've got these various things going on. A rate of the actual transcription, but then we might have hydrolysis, we might have precipitation, we might have formation of double-stranded, and so on. And actually, by thinking through the mechanisms and looking at experiment design, we're actually able now to create, kinetic parameterized models to give us quite a high level of accuracy. And we've been fortunate that within the projects in our hub and some projects funded by Wellcome Trust and collaboration with King's College, unfortunately, our main collaborator is unwell, so he couldn't be here today. We're able to get a very orthogonal set of data. So this is showing the kind of sequence richness in the data set that we have in these 800+ experiments with different so-called constructs. Constructs means the protein that you're coding for. And we're looking at, medium-sized RNA and large-sized, typically self-amplifying RNA, and different sort of titers. So we've got a very rich data set, which allows us to really test our models. So I think what we found in the last 15 years is these systems methods, they really benefit from collaboration with people who really know how to do experiments and really know how to do analytical science. So, you can't really get away without that. So having gone through this process of model building and parameter estimation and so on, we started to look at, how would this work as a platform, and also how do these advanced methods actually help us? So the first thing we found is once we've built this understanding, we have 15 parameters, but actually by doing sensitivity analysis and also understanding from the biology and the physics of what's going on, we really can come up with this concept of a platform technology where you can essentially not repeat a design of experiments associated with 12 of those parameters. In other words, 12 of those parameters of the model of how RNA is produced in this complex process don't change whether you're making influenza vaccine or rabies vaccine or anything else. So they're not construct specific, they're only platform specific. So once we've built a model for the platform, that actually already exists. So all we need to do is every time we have a new RNA to produce and we want to say what's the best way to run this IVT process, we just need to reparameterize our model for these 3 parameters. We only do relatively few experiments. Now, experiments. So there was a very nice talk in the student and postdoc session, the very last talk, where he explained that model-based design of experiments, which is information seeking, is much more powerful than just maybe classical approaches or exploitative approaches. So here we're looking at the design space. So the design space is the area within which you want to operate to make sure you get a high-quality product. So what I'm showing you is a probabilistic design space. In other words, our models are not perfect. They have some uncertainty in them. And that uncertainty is reflected in how confident we are in our design space. And so the blue is highly confident, and red is we're not really confident. And if you choose the right experiment design, you can be very confident. And that's what I'm trying to show here. So this is what we call a screening design. So if we take a new construct, so in this case it's a construct for a protein that just gives a kind of illumination. The classical design is what we call a screening design, where if you've got 3, remember I said there are 3 parameters that are construct dependent. So screening design, you might, look at some extreme points and a midpoint. So you do those experiments, which we did, and then you put them into the parameter estimation process, and then you calculate your probabilistic design space, you get this. So you've got a reasonable confidence you want to operate here, but you've still got a lot of uncertainty. If you use a model-based experiment design, which is designed, to optimize information or to, minimize uncertainty in your parameter estimation, so this is what we call a de-optimal parameter design. Interestingly enough, it puts the points in a different kind of point. We end up over here, here, and here. So our 3 experiments are not what you would do in a classical DOE. But the model has said, if you run these 3 experiments, they are maximizing the sensitivity of your outputs, which are then used to define your quality attributes with respect to these kinetic parameters. And so when you estimate the kinetic parameters, you'll get high confidence. So by doing the 3 orange experiments, we get a much nicer design space. So we're very confident we can operate in these areas. How good are the models? So the models, we feel, are pretty accurate because they're capturing very complex phenomena like the titer. So this is the prediction versus experiments for the titer. But then integrity, there are no kinetic models in the literature that predict integrity, but we're able to predict integrity very well. And for the production of double-stranded RNA, again, very complex process, not well understood. Often people actually measure their titer wrong because in the titer they're actually including the double-stranded, so they're getting the wrong titer. So here we're actually able to capture very well the production of this double-stranded RNA. So that then allows us to develop quite sophisticated design spaces where, for example, depending on what your criteria are for integrity. Now, one thing I should say is, we don't need perfect integrity to get a good efficacy. So eventually we need to tie these design spaces into how well does this actually work in the patient. But here we see our 3 important construct-specific parameters, and depending on how high I put my integrity, I have a certain design space, and then depending on where I operate, I can be confident in a certain titer as well. So we can start to go from a disease construct to tell someone who's got a manufacturing facility, please operate, with this much magnesium, this much of your NTP concentration, and this much of your polymerase. And they can be confident to go away and do that. So instead of having to do hundreds of experiments, you can actually meet this 100-day challenge. Another interesting thing around model-based experiment design is if you want to do some experiments also to demonstrate how well the process will work, you would do a different kind of experiment from the experiment that says I want to maximize information or want to minimize parameter uncertainty. So here we were doing the case of we want to do 3 experiments that tell us the most about the range of possible outcomes of two metrics, R titer and the degradation. And so, you know, again, this was our possible design space. It would be very, very difficult to do a, let's say, classical experiment design to say which 3 points in this design space should we use that will give us the most information about these KPIs. And it turns out, you know, if we do these 3 experiments in the input space, we get these 3 outcomes in the output space. And what's particularly interesting about this process here is when you look at the behavior, particularly of some of these inputs, in the detail, there's some non-monotonicity. So the relationship between the process parameters and the output are non-monotonic. So if you think of some of the classical approaches, like response surface maps, those are essentially constants plus linear terms plus quadratic terms. They can't capture non-monotonic behavior. So they wouldn't give you good experiment design. So you could do model-based experiment design using an RSM, but you wouldn't get the same outcomes you get with the approach that we are following. So very briefly, my final case study, global supply chains versus local manufacture, which I'll keep very short, because this is something which people talk about a lot, but they talk about it very qualitatively, right? And it comes about because of this issue of sharing of IP and sharing of vaccines. So what we're looking at here is really, you know, under what circumstances does it make sense for every country to make its own vaccines, or every region to make its own vaccines, versus, you know, have a centralized facility? And so we built on our knowledge of these different manufacturing models and different manufacturing strategies, and we looked at some things like, let's say you made it all in the USA, and you shipped it around the world, versus you make things in Kenya or Ethiopia, and so on. And what you find is that, you know, for most technologies, the most profitable or equally the lowest cost is the black bar, which is, you know, just make it in one place and send it around the world, because you get economies of scale. We didn't even look at secondary things like skilled people, supply chain of reagents, because there are a lot of reagents that you need. We're just looking at manufacturing supply chain. The one thing that does make distributed manufacturing interesting is when you have things like RNA that's made in a so-called cabinet-contained, relatively small-scale process. And the reason for that is that when you're making pharmaceuticals, you need to build facilities that are super clean. So you need amazing air handling, water treatment, other utilities, containment, biological containment, and so on. For conventional processes, the whole facility more or less has those characteristics. The so-called cabinet-contained process, which only works for things like RNA because the doses are small, so in a small facility you can make millions of doses, the whole facility can be made to some basic level of cleanliness. It's only the cabinet where you're doing the manufacturing that needs to have these sort of aseptic properties, and then it starts to be something that actually could work well in a distributed manufacturing. You still need to think about the supply chain issues, like how will you get all those reagents in the right quality to all those places in the world, rather than just, say, to New Jersey. So lots of interesting things still to do there. Lots of open questions as well. So, as I said, the RNA, it's coding for a specific protein. But if you look at the SARS vaccines, they don't just copy the exact genetic code for the spike protein. There are lots of clever things people put in there. For example, you can have a slightly tweaked 3D conformation of the protein, improve the stability, efficacy. So there are clever things you can do there. So how do you codify that protein design, and can you ultimately optimize it? Once you've got a protein sequence, so that's an amino acid sequence, we have this other feature in biology called codon redundancy, or which allows us codon optimization. So if you remember your biology, you've got the 3 bases will code for an amino acid. But every amino acid has multiple triplets of bases that can code for it. So there's not a unique way of coding for an amino acid sequence. So there are some good rules of thumb for which so-called codon sequence, your 3 bases, you should use for each amino acid. But actually there's a lot of opportunity to optimize that sequence. And actually, optimization, what does it even mean? Is it for the transcription process, that's the making of the RNA, or is it for the RNA translation basis, what RNA is doing in the body? There's a lot of interesting work going on in modifying the basis. So those individual components that make up the RNA, you can tweak the chemistry, especially on the sugar part, and you can do it in such a way that it, for example, is much more stable to hydrolysis, or it might work better in the human body, and so on. And in fact, there was a particular base modification which meant that it really worked rather than was a theoretical thing for which the Nobel Prize was won. But when you do this, you might get some funny things like what's called frame shifting, which is when you are making the RNA off the DNA, it gets lost by one position and it gets in a mess and you end up with something that doesn't work. So codon optimization is really interesting because, these biological systems, they're inherently digital, right? It's just a 4-letter sequence that repeats in different ways. And then there's the, 20-something amino acids that make up the proteins. So it's inherently digital, and that allows us a lot of space for machine learning and maybe other techniques. There's a lot of formulation optimization. I find working in formulation science is fascinating because they come from typically a chemistry background, and it seems like almost a kind of alchemy. Like they have all these ideas of different lipids, different constructs and so on. And so there's so many different things you can do there. Lipid nanoparticles, polyplexes, polymer encapsulated LNPs. So there's a lot of chemistry optimization that can be done there. There's a lot of processing optimization, I think, that can be done in formulations. And, RNA at the moment, it needs to be stored relatively low temperatures. So, of course, making IP-free, heat-stable formulations would be really good. New process designs like circular RNA, so then, again, has greater stability. New chemistries for the individual elements, new therapeutic areas, so non-vaccine applications, cancer applications, protein replacement applications. And then an area which I think might be worth exploring is peptides. So peptides, as you're probably all aware, is an incredibly fast-growing therapeutic area. So these 30 to 40 amino acid peptides are selling extremely well for appetite suppression, diabetes, and the like. And they're very difficult to make because they're made step by step in the synthesis process without a template. So you've got to, add one amino acid at a time and then remove it and so on. And it's a complex build process. So, looking at RNA and learning how you produce a templated product, is there something like a cell-free translation process that could rapidly make peptides 100% correctly because you're making them off a template? So let me conclude. Vaccines has a lot of interesting integration between the product design, like the vaccine sequence or the vaccine product, the process design, so lots of other things that I haven't spoken about. One interesting thing that my colleague Karen is working on is yeast to make proteins or VLPs. And if you can encourage the yeast to secrete the product into the extracellular medium of the fermenter, the separation process would be absolutely simple. So that's a case where re-engineering the organism to maximize its secretion completely changes the way you do process design. So these things interact with each other. Lots of new developments in biotechnology. So biotechnology moves incredibly quickly, and for us in manufacturing, we struggle to keep up. So we really need to stay on our game. And that's where we see this modeling can be really important. I think especially these ideas of trying to separate out the platform-dependent aspects of the model and the product-dependent aspects. So really good opportunities for interdisciplinary collaboration. I'm so pleased to see many collaborators here today, so I want to say thanks to many of them. So people at the top, they're all the people that have been directly involved in some of the work that I've discussed today, going back some years. I think particularly special thanks to Cleo, who's also going to give the vote of thanks, but who's been collaborating with me on this topic, and many special thanks to Robin too, who works with me in the Future Vaccine Hub. And then, of course, very special thanks to Costas and Roger. You know, I wouldn't be doing this talk if it wasn't for them, because, you know, it was from them I learnt so many of these methods, but also, you know, just ways of thinking about problems. Because I think probably the most important thing, hopefully everybody takes away from the Sargent Center is, you know, systems thinking. You know, how to think about problems and structure them, and then deal with them in a systematic way. So thanks very much. I'll now pass to Claire.
Thanks very much, Nile, for this insightful tour of PSC, the impact of PSC in vaccine manufacturing at all scales. I think we saw every scale. And now I'd like to invite Professor Cleo Contoravdi to give the vote of thanks. Thank you.
So let me start by thanking Claire for the opportunity and the honor, really, to give the vote of thanks, because I'm also an alumna of the department from the undergrad and the PhD, and it's really been a pleasure and an honor to have received immense mentorship from Nile, and I think I speak for a lot of my colleagues here today in that he has really defined our career trajectories, it would be worth saying. And listening to his talk, what strikes me is actually how early Nile started working on biomanufacturing. King Hei, Trevor Chang, was my contemporary. He started working on the influenza vaccine supply chain problem in 2002. So a long time before any pandemic, a long time before any one of us was thinking, perhaps except for Robin, was thinking about disease X, or how we would respond to an emergency, a global healthcare emergency. And although it started with traditional, if you like, supply chain management problems or capacity planning problems, which were commonplace, if you like, in traditional process systems applications, I think it would be worth saying that I see Nile as a true pioneer in terms of thinking how process systems engineering tools could fit into an industry that's so heavily regulated and where computational tools are still today not finding routine application. So I wanted to spend a little bit of time, just a short amount of time, to talk about the evolution of going from our bread and butter, so the supply chain management, the capacity planning under uncertainty, to today, to the work that Nile has been doing as part of the Vaccine Hub, which he set up with Robin in 2018, again, really ahead of the pandemic, starting to think about issues that hit us so fast, only 2 years after establishing the hub. And what is evident from what Nile talked to us about today is this evolution from going into supply chains and capacity planning to actually drilling down on specific unit operations, being able to describe them clearly, simply, and in a way where we could collaborate with experimentalists to develop solutions that had a real impact in industry and for our collaborations around the globe. I wanted to really emphasize that the Vaccine Hub has a clear mission to help manufacturers around the globe, and primarily in low and middle income countries, to establish or even progress their existing vaccine manufacturing facilities. And in this sense, the work that Nile has presented today has two clear benefits. First of all, the optimization of vaccine manufacturing technologies in order to make them cost-effective and to make vaccines accessible to populations around the world. Being in the UK, perhaps we don't realize how lucky we are in having access not really to the COVID vaccine. People have many thoughts around that, as Nile correctly pointed out, but to routine childhood vaccinations. So being able to employ process optimization to work closely in the way that Nile has this remarkable ability to really take a really complex notion and drill down to the details, explain the biology in simple terms, and then translate it into a problem that we can all solve to make actual real impact on process optimization and on the cost, the final cost of the vaccine in a low and middle income country. And finally, I just wanted to talk about RNA vaccines and Nile's advocacy for this pre-qualification of the RNA platform. He's worked with the Coalition for Epidemic Preparedness Innovations and with the UK government. And all of the things that this means in terms of really trying to reach vaccine equity. So in closing, I'd like to say that Nile started by saying about Roger Sargent's clear thinking and brilliance. And I think that what Nile demonstrated us today is these are qualities that he himself possesses. I think anybody who has worked with Nile will know that he has a remarkable ability to understand really complex problems in biology, even better sometimes than biologists themselves, to explain them clearly and to really articulate the added value of process systems engineering tools in operations that range anything from energy systems that have clear relevance to process systems engineering all the way to vaccines and to biomanufacturing. So I'd like to close by thanking you, Nile, for the thought leadership and the mentorship.